this video we will discuss about these NK cells are found in the low fibroblast region of the tumor microenvironment of pancreatic adenocarcinoma. In the previous video we have understood that NK cells are found in the epithelial regions of the tumor microenvironment of pancreatic adenocarcinoma. However, the hallmark of the tumor microenvironment of the pancreatic cancer is the high cancer-associated fibroblast region. So they wanted to check whether the fibroblast regions also contains the NK cells or not. So first what they did is they identified the region of interest where fibroblasts are found, and they made them into the 4 sections or with different level of fibroblast. That is shown in the panel A, where we can see that the quantile 1 has low amount of fibroblast and it increases with increasing the quantile. And when they check in this respective region of interest about the amount of epithelial cells and the NK cells, they found a decrease in the epithelial cells and the NK cells with increase in the fibroblast. That can be seen in the panel B and panel C respectively for epithelial cells and NK cells. And after that, they also check the collagen, which is usually formed when the fibroblasts are formed, and they are able to see that increase in the collagen. So this results, that is panel relationship between panel A and D, is in line with the with the relationship that is available in the literature. That is, fibroblasts are usually formed and cause increase in the collagen 1. And after having the informations that NK cells might be low, NK cells will be high in the low fibroblast regions, they check the immunofluorescence image of different immune cells and this NK cells. That is, they check the alpha SMA, ECAT, PANC-CA, NKGT2, and what they are able to find out in both of this is that the high fibroblast regions seems to have this low presence of the NK cells. That is, we can see that NK cells are not present close to these high fibroblast regions. Fibroblast regions are usually represented by alpha SMA, and this is also represented by alpha. Here we can see the alpha SMA does not seems to be overlapped with the NK cells. This is the zoom in image in the panel F, where we can see that the NK cells seems to be surrounding the collagens but not within the these alpha SMA signals or the fibroblast. And same things, an amount of NK cells seems to be higher in the Q1, but it is decreases as we go with increase in the fibroblast. And these things are fibroblast abundance quartile is true also for the other immune cells, where we can see that with increase in the collagens, the immune infiltrations goes down, as this fibroblast act as a physical barrier to prevent the immune cells to prevent the immune cells to attack the cancer cells, or act as an immune evasion barrier. Thereafter, they check the different quartile of the fibroblast and they plot the heat map based on the different relative expressions of the different immune cells markers. And what they are able to find out is that this collagen 1, which should be higher in the they are able to see that in the case of this quantile 4, they see a low presence of this apoptotic markers such as cleave caspase 3 are low, and whereas in the stroma we can see that high presence of these markers and the collagen 1 is also higher. Whereas the quantile 4, the presence of this apoptotic marker seems to be lower compared to the case of this quantile 1, 2, 3, indicates that with increase in the fibroblast, they see a low apoptotic of their cells, and it does a job of immune evasions better with increase in the fibroblast. And in the different quantiles, they also check different immune cells markers such as CD69, CD107, alpha, granzyme B is the cytokines that are released to trigger apoptosis, and IFN gamma also helps in the apoptosis, and those things does not change too much in the stroma quantile 1, 4, and thereafter they did their final analysis. In this they did the final analysis. We can see that in the region they did the final analysis in the region of in this the panel G is about the fibroblast quantile and fibroblast different quantile 1, 2, 3, 4, and panel L is about this panel L is about this NK cells, where NK cells are found more. Panel L is about the quantile where NK cells the NK cells are found, and here we can see that the NK cells in the quantile 1 in the case of quantile 1 also we can see a decrease in the apoptotic marker, whereas in the case of quantile 1 we can see a increase in the apoptotic marker, which also indicates that there is a high apoptosis occurs where NK cells are higher, and in the case of this quantile 4 we can see the apoptotic markers goes down. This indicates that the NK cells has a direct role towards triggering apoptosis, as when the NK cells goes down, the apoptotic factors also goes down, such as cleaved caspase 3, and the proliferation markers also goes down. Whereas and similar results, the panel L results are similar to that of panel G. So both of this indicates that the fibroblast has a role towards immune evasions, whereas NK cells has a role towards immune suppression. And NK cells are found in a region where fibroblast amounts are lower. So NK cells can also prevent this fibroblast can also prevent NK cells infiltrations. So I hope you understood. In brief, this seems to be a little bit confusing, but you might have understood the overall idea of their results, or overall conclusions of the results, that NK cells are usually found in low fibroblast region in the tumor microenvironment of the pancreatic adenocarcinoma. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.